All right, well, thank you and welcome to Scalable Interactive Graph Genome Visualization with Pantograph. I'm really looking forward to presenting this work from a really terrific group of people. It was a group effort that we got done with in our quarantine time. We were tasked by the 1001 Genomes Project and the Max Planck Institute, among others, to really tackle some foundational problems in comparative genomics. And it really all comes down to alignments. So alignments are the basis for genome assemblies, finding transposable elements, looking at within species variations, which we have to align a bunch of different de novo genome assemblies, looking between species at conservation, phylogenetics, functional annotation. All of these rely on alignments. And so the way that we do alignments is very important. In particular, when we do reference-based alignments, it's very common to throw away structural rearrangements. And that accounts for about half of all the biologically meaningful information. So what's the problem here? Well, originally we had 100 genomes, and now we have 100 genomes for every single study. The 1001 Genomes Project with Arabidopsis thaliana looks at putting together 1,000 de novo assemblies. Of course, we have the 1000 Genomes Project with human genome assemblies, and now we're looking at 1 million human assemblies and many more projects to follow. This kind of computation already was taking weeks with even 2 genome alignments. And so now we could be looking at years with the same tools. So that's just not feasible. And this reference bias problem, it becomes worse with more genomes. So for example, the human genome reference is a European. If you use a European reference to compare 2 Africans, they look more dissimilar than if you were to use an African reference genome. And this reference bias problem applies all across the field of comparative genomics. So it'd be great to have a tool that doesn't have this reference bias problem. Alignments require many different parameters, which require tweaking. And so it'd be nice if we made that process more transparent. And we'd like a tool that allows us to summarize so we don't have information overload, to zoom out to chromosome levels and look at the big picture without getting drowned in detail. And we like a data format that doesn't become unwieldy when we have rearrangements on top of rearrangements. And because of all of these problems, currently we have many data analyses that are just throwing out structural rearrangements. So why does that matter? Well, researchers need better tools. And hopefully your research matters. Let's take the case of COVID-19. Right now, we have probably billions of individuals infected with, well, trillions of copies of a viral genome, each producing their own mutations. Now, normally that wouldn't matter for the mutation rate, except if you do a selection sweep. If we come up with a vaccine or drug or anything like that, and we begin eradicating the virus from billions of individuals, that creates a selection sweep which specifically selects for resistance to whatever our solution is. And so if we don't know about that variant, we can end up creating pandemic 2.0. We could have COVID 2021. And so we would like to use Pantograph to help monitor for the existence of these other variants and to keep on top of that. And that means not throwing away any kind of variant: SNPs, insertions, deletions, and structural rearrangements. So let's walk through a simple example. At the top, I have a multiple sequence alignment with 5 individuals and a green block of text. This is a syntemic block with a pink element at the beginning and at the end because of rearrangements. Now, multiple sequence alignments don't really allow you to show this, so I've shown it with coloring. But if we go down here to our pangenome schematic, each row is still one individual. And if we start on this second row here, you see you read it off TCCAA. And then when you hit this pink block, you follow the pink link over and down until you go and land on the second row again. And then you read GG. You skip this T because it's not present in this individual, and this T is present. And then you follow the second green link up to right here. Second row again, read across. There's a snip with the trivariate site, and across to the third structural rearrangement, which just skips that. And then we have GCTAT. And you can see right up at the top, same sequence, but this allows you to show it in any order. And so you see the first and the third, they read straight across, left to right. And that's because with rearrangements, there's no single global sort order. So we need this sort of thing. So this effort was part of a much larger virtual biohackathon, and we've made available through the public sequence resource team 1,300 viral genomes, which you can access at this URL. You can also access this through Sparkle queries, which can integrate with many different kinds of metadata and annotations so that you can ask more biologically meaningful questions like, are the active sites of a particular gene conserved? And we're hoping that this is meaningful. I encourage you to check out the talk and check out our live demo of the website. All right, thanks.